Hey, you came back. Well, here in this segment, we actually look at what happens during photosynthesis. How do you take that light energy and convert it into sugars? So, um, the notion is you need to take energy and convert it from one form to another. We start with light energy from the sun, and then we need to turn that into the chemical energy ultimately of sugars. Okay, so we have some sort of intermediary steps we'll get to, but that's the goal. Is you convert light energy into chemical energy and sugars, and then you can take those sugars, send them on over to the mitochondrion, and then break those down and make ATP. All right. So the structures or the molecules involved in the light reactions. Light reactions are so named because they require light. They're embedded in this thylakoid membrane in the chloroplast. And the big thing to do is follow what happens with electrons in this process. All right. So if we take a look, here is the thylakoid membrane. This is in the chloroplast. Okay. So if we bump back here a little bit, you see the green leaves here. Here are the chloroplasts. And if we zoom in on a chloroplast, it's got these green membranes, the thylakoids. So now we're going to zoom in on those thylakoids even more. And then this is looking at a thylakoid membrane. Now, embedded in the thylakoid membrane are important protein complexes called the photosystems. So there's photosystem 1 and photosystem 2, or PS1 and PS2. Now, the photosystems are not named in the order in which they occur. They were named in the order in which they were found. So that's a little confusing. But the big idea is if you look at those photosystems, you see the little green dots. Those green dots represent the pigment chlorophyll. So chlorophyll is a photosynthetic pigment, and we know pigments can absorb light energy, right? So what happens is light strikes the chlorophyll pigments, and it excites electrons. Electrons get moved up to a higher energy level. What happens is a special pair of chlorophyll molecules have their electrons excited, and they actually release those electrons. Those electrons are captured by electron carriers, and they actually travel through an electron transport chain. So if you recall the old electron transport chain from cellular respiration, this is a different adaptation of electron transport chains. So again, light strikes the chlorophyll in photosystem 2. Electrons get excited, and then they're passed along an electron transport chain to photosystem 1. Photosystem 1 then has those electrons excited by more photons of light. So those electrons get excited, and they again get passed on to a little molecule that shuttles the electrons, ultimately to an enzyme called NADP plus reductase. So, reduction is a gain of electrons, right? So what it does is it passes on electrons and then associated protons to a molecule called NADPH. So NADPH is another electron carrier. If you recall back to the cellular respiration chapter, there was NADH and FADH2. Okay, this is sort of another vehicle. This is another electron carrier, NADPH. And then the NADPH will be used for later processes. So this is all well and good, right? You have energy from the sun exciting electrons in a pigment. Those electrons get passed along to another chlorophyll molecule. Light strikes that, electrons are excited again, and their electrons and then associated protons are ultimately passed on to this electron carrier, NADPH, that we'll talk about later. Now, there may be a little bit of an issue you notice with this if these chlorophyll molecules lose their electrons. If you lose electrons, hey, you got to replace them, right? So what's a good candidate for electrons? Let's use water, okay? So this is why plants, in part, why plants need water. This is why they need it for photosynthesis. In photosystem 2, if these chlorophyll molecules lose their electrons, they need new electrons. So what will happen is water will be split, and then the electrons from water can be given to these chlorophyll molecules in photosystem 2. So the electrons are removed from water and passed on to chlorophyll. The protons then will be released into this area called the thylakoid space, And ultimately, these protons will be released from the cell or released from the thylakoid and can join things like NADPH potentially. And then the oxygen is released as a waste gas. So the oxygen we breathe that we need to live, that oxygen is produced here inside the thylakoids when water is split. So splitting water gives electrons to the photosystems. It releases oxygen as a waste gas, and then it releases protons into the space, and then the protons will be used in other ways. Okay? So one thing that's cool about this is, let me move this over. These protons from water, when they are in the thylakoid space, notice how they're encapsulated in a membrane. What that does is create a gradient. So protons get trapped inside the thylakoid space, but look at the exit here. Hey, we've seen that before. ATP synthase. So when protons pass through ATP synthase in the thylakoid membrane, ATP synthase spins. It brings together ADP and an inorganic phosphate. They join up, and boom, you've got ATP. So that's sort of a fascinating sort of sidestep in this light reaction process, is that when you split apart water, you release the oxygen, but then you can use the protons to create a gradient to power some ATP production. So when it's all said and done, what the heck has happened? Well, with the light reactions, what we've done is taken light energy and converted it to chemical energy in the form of NADPH and ATP. Okay. Now, to replenish electrons lost from that, we split apart water, right? And when we split apart that water, that creates the oxygen, molecular oxygen in the atmosphere that we breathe. So now, at the end of all of this, what have we wound up with light reactions? NADPH and ATP. We've converted light energy into the chemical energy of these molecules. So now what I'd like to do is talk about what we do with that chemical energy. All right. So the energy from ATP and NADPH is used in another set of reactions called the Calvin cycle, named after Melvin Calvin, the man with two first names. Or sometimes they're called the dark reactions or the light independent reactions. Again, however they call it on the AP exam. This happens in the stroma, that fluid in the chloroplast. So here, sort of backing out, we've got our chloroplast. The thylakoid membranes are where the light reactions happen. That's where the photosystems are embedded and ATP synthase is located. The ATP and NADPH get released into the Calvin cycle. And basically the enzymes for the Calvin cycle are in the stroma of the chloroplast. But what they're going to do is use this energy, this chemical energy in ATP and NADPH, to take carbon dioxide, rip it apart, and rearrange it into sugars. All right, so how does this happen? One thing I want to mention is that you don't need to memorize the names of these molecules. Just sort of pay attention to inputs and outputs. I'll mention names just so that we can have some specificity, but you don't need to, know all these intermediates or anything like that. Just think about inputs and outputs and basically what's happening. So there's atmospheric carbon dioxide. What we have to do is first what's called fixation. You take atmospheric CO2 and then you attach it to some organic molecule. Okay? So what happens is CO2 is joined to an existing molecule. It's called RUBP. It's like ribulose biphosphate. But what happens is an enzyme, it's called Rubisco, takes that atmospheric CO2 and joins it to RUBP. And then we've got this 6-carbon molecule. Okay. Now, this is where our ATP and NADPH from the light reactions come into play. You use the energy and the protons and electrons from NADPH and ATP to rearrange these molecules into these G3P sugars. And then what happens is the Calvin cycle spits out these little 3-carbon sugars that can join together and form glucose. All right. So I think that's what we're talking about here. The first step is fixation, where you basically attach the carbon from carbon dioxide to RUBP. The second step is called reduction. Again, reduction is a gain of electrons. So you take the electrons from NADPH and add that. And again, you use some energy to rearrange these molecules, and you end up spitting out these 3-carbon sugars, the G3P sugars. that ultimately can combine to form glucose. Now, when that happens, you're going to be spitting out 3 sugars at a time, but you can take some of the existing molecules, rearrange them, and then that recreates RUBP, and that's why it's cyclic. And again, that's what we get into with step 3, regeneration. With regeneration, you use a little bit of energy, a little bit of ATP to rearrange these molecules back into RUBP, so Rubisco can basically fix more carbon from carbon dioxide. All right. So big picture, I think this image is good. Big picture, what we have going on is light energy is converted into the chemical energy of ATP and NADPH. That happens in the light reactions. And if we zoom in, here are the light reactions in the thylakoids. So there we see it right there. Light energy strikes the pigments, the electrons are excited, pass along to NADPH, and then you use water to replace those electrons that were lost. The protons from the split water power the production of ATP. So at the end of the light reactions, we've got NADPH and ATP. Then in the Calvin cycle, that NADPH and ATP is used to take molecules created by carbon from the air, rearrange them into sugars, and then you've got your sugars, okay? And then you can take those sugars and send them on over to the mitochondrion. That'll break down the sugars and make ATP. Whew, that's a lot of steps to make some ATP. All right, so that completes this segment, and then, of course, we'll finish up with like all the oddball things that can happen.